Thanks, Susan. Thanks, and good morning to everybody, and thanks to the organizers for allowing us to present our company and data at this meeting. So I'm going to introduce you to Ocunexus Therapeutics and our drug Xiaflam, which is an oral small molecule, which is used to regulate the inflammasome very far upstream, so we're inhibiting NLRP3. I guess you can all recognize this mob here. This is the Monty Python group, and I'm going to show you something that is completely different today. So we are a clinical stage company with transformational upstream mechanism of action, controlling the inflammasome. We have a high potential, therefore, of modifying the course and the treatment of GA and diabetic retinopathy. We have an orally administered once-a-day drug, which is a connexin 43 hemichannel inhibitor. We have multiple phase 2b studies, which provide us with a robust human safety database, showing good tolerability and no safety concerns. We are ready to file the IND by the end of this year and have Xiaflam in the clinic for GA and diabetic retinopathy by Q2 next year. We also have an extensive and strong patent portfolio through 2040. Our drug target is the connexin 43 hemichannel. The hemichannels are continuously forming in the cell membrane, and the hemichannels remain closed until they dock with another hemichannel in an adjoining cell, and this is to create the gap junction, and this is occurring in just about every organ in our bodies. In disease states, the connexin 43 is overexpressed and prematurely open in the cell membrane. Open undocked hemichannels become pathological, allowing intracellular ATP to move into the extracellular space. And this is the signal to start up and recycle the inflammasome pathway of inflammation. This is a diagrammatic format of the actual pathway that we're targeting. And the place to really consider the most dangerous is the one in the lower right, where we see the recycling of the inflammasome. So the inflammasome will continue to recycle, continue to activate multiple pro-inflammatory cytokines, and then have downstream pathological effects. And the way to stop this recycling is to intervene far upstream, and that's what our drug is going to be doing. In order to look at the mechanism, we looked at cellular assays, and this is on the left-hand side is looking at ATP release. And what we can see in the black bar, when we challenge these cells, these are retinal pigment epithelial cells with TNF-alpha and interleukin-1b. The black bar shows the basal level of ATP, and you can see the green bar, there's significant increase in the ATP release. When we add a hemichannel blocker, which is the purple bar, it knocks it back down to baseline. And once that ATP is released, we see the inflammasome on the right-hand side being activated. So the inflammasome is assembled, and we can actually measure it and quantify it. And you can see again in the baseline, if we look at NLRP3 spot count, it's almost zero. And then when we look at the ATP activated cells, we get a huge increase in the inflammasome activity and assembly. We then give it a hemichannel block, and it goes right down to baseline again, almost to zero. And then to show the mechanism, we add exogenous ATP, and we can restart the assembly of the inflammasome. So we've proven our mechanism for sure in cellular assays, and we've also looked at human donor retinas, which are shown here. And these are retinas with diabetic retinopathy and macular degeneration. And the green stain is the connexin 43, which has been stained, and you can see overexpression of the connexin 43 in these donor retinas with the disease compared to the normal on the left. We've then taken the drug, the oral drug, into the acute bright light model of GA, which follows the course of geographic atrophy starting in the choroid and ending up with destruction of the retinal pigment epithelium. These animals are left in bright light for 24 hours, and the oral drug is provided to these animals 2 hours into the light exposure period. One group is the Xiaflam treated, and the other group is placebo. And what you see on the left-hand side here is an OCT of the retina in the treated eyes, and this is 3 months after treatment. And you can see a full thickness retina with all the layers well described. And on the right-hand side, you can see the non-treated eyes or the placebo-treated eyes at 3 months, who have almost complete destruction of the neural retina and the RPE, and a very thin retina. And if we look at the ERGs that were carried out below that, in the treated eyes, we have a full visual function continued for 3 months and completely extinguished function in the eyes that were placebo-treated at 3 months. We've also come across an isolated strain of Sprague-Dawley rats, which was serendipitously found in Professor Green's lab in New Zealand, which developed real diabetes within 4 weeks of birth. And these, when we looked at OCTs, actually have clinical signs of diabetic retinopathy. So within 4 weeks of birth, they are showing micro and macro aneurysms. So this is a chronic model of real diabetes, not something that's been induced. When we do the OCT imaging, which you can see over here, if you look at the top, that's the normal Sprague-Dawley rat. And then if we look at the diabetic eyes, you can see the micro and macro aneurysms. So what we did was we took the one group and split them into two, and the one group was treated with the oral drug, Xiflam, and the other group with a placebo. And you can see in the top OCTs, you can see the microaneurysms on the left, and you can see regression of the micro and macro aneurysms 4 weeks after treatment. And the same thing happened with the visual function when we did the ERGs. You can see the treated eyes actually got restoration of vision as the retinopathy started to regress, whereas the non-treated eyes actually continued to decrease. We have 2 trials. The one is for geographic atrophy, it'll be a 12-month trial, and we'll be looking at stopping progression of the lesions in GA and regression of GA lesions if we are successful. In DME, we'll be looking not only at the DME, the corneal thickness, but also at kidney function, since we know that chronic kidney disease is associated with the inflammasome, and these are patients that are going to be getting a systemic drug. So we will look at kidney function as well. So what is the value proposition? We are phase 2B ready. We have the disruptive potential for disease modification, preventing the recycling of the inflammasome, versus the current treatment paradigm, which is intravitreal injections for the recycling of flare-ups of the disease from monthly to PRN for life. We have an orally administered drug, and we are looking to close around, and we have long patent life on the drug itself and the mechanism. These are our medical advisors, pretty good group of people who are helping us to move through the development. And we are leading the pack. So when it comes to inhibiting the inflammasome and being upstream, regardless of whether we're treating ophthalmology or diseases in CNS, for any disease where we are looking at the inflammasome, we are ahead of this curve. Thank you.
Excellent.